Hello, my name is Dr. Taisha Rowland. I am a director of a stem cell research center at the University of Colorado in Boulder. So the name of the center is the Stem Cell Research and Technology Resource Center. And basically what it is it's a shared facility for researchers to come do stem cell research. So what's behind me right now, you might be wondering what my background is. This is actually what's called a stem cell colony. So it is the big cluster here, that's a couple hundred cells, and they're specifically human-induced pluripotent stem cells, or they look the same as human embryonic stem cells, and they always grow in a colony like this of a couple hundred cells. Surrounding them are other cells. So you can see around the edges there, those are other cells. They're what's called feeder cells, and they basically just support the stem cells. So a bit about myself, I have a bachelor's degree. Is specifically in molecular, cellular, and developmental biology, or MCDB. After receiving my bachelor's degree, I went on and received my PhD at the University of California in Santa Barbara. And this was also in molecular, cellular, and developmental biology, or MCDB. After that, I did a couple postdocs. A doctorate is your PhD degree. A postdoc means you already have a PhD, so that's why it's postdoc, or postdoctoral studies. So people who have a PhD, they'll often go and do more research to become more qualified. But after that, then I became the director of the shared facility where I'm at now. So if you want to be a professional stem cell researcher, or professional researcher in general, a PhD will let you do just about anything you want to do in the field. So you can get a master's degree, or you can get a bachelor's degree. What it really means, though, is that down the road, you'll probably be reaching a ceiling at some point. And if you have a PhD, that's an additional qualification that will let that ceiling be a little higher for you. For my position, the requirement is to have a PhD or a master's. So master's might get you in the $40,000 to $60,000 range, whereas if you have a PhD, it might be more around 60 to 80, 85,000. So what this facility is, as I said, it's basically a big lab space that is dedicated for doing stem cell research. So it is a shared space, and other researchers can come in and do research here. So here are a couple pictures showing basically, what the space looks like. You can see there's a lot of space. It's clean. People come in to do work with these cells. One of the things that I've been asked to share is sharing a time that I failed and how I learned from it. So these cells are very technically challenging to grow. At this point, I have about 10 years of experience growing these cells in the lab. So I've made just about every mistake there is. And every time I make a mistake, I learn, oh, that's what it looks like if you make that mistake. And so the next time, if I see the cells looking like that, or if I'm helping someone else and I see the cells look like that for them, I can say, oh, it's probably this that happened because it's happened to me. And that's how they looked. And so it's all a learning process, and it helps, it makes it easier to troubleshoot. The more that you do something, the more you know when something goes wrong, what it looks like. And then that helps you figure out, what you should change. So what's behind me here is what is called a biosafety cabinet. This is basically a piece of equipment that we use to take care of our cells. So whenever we have to change out the liquid on the cells, we use this environment. And what I mean by changing the liquid, we call it media. And the media is basically, it's the food for the cells. And so every couple days, we usually come into this biosafety cabinet and we remove the old media and we put fresh media on it. So the reason it's important to use a biosafety cabinet when we are working with the cells is that they need to be in a sterile environment. And what sterile means is that the cells don't want to be exposed to any bacteria. And this is because the bacteria can kill them. So this biosafety cabinet is a way to be able to work with these cells in a sterile manner. You can see I'm also wearing a lab coat and gloves. And this is important, again, whenever working with these cells to be sterile, to keep everything really clean. So the math problem that I wanted to share today is kind of an example of a math problem that I use a lot in the lab. Is a classic equation called C1V1 equals C2V2. C stands for concentration and V stands for volume. The C1 is the initial concentration, V1 is the initial volume, C2 is final concentration, and V2 is final volume. The way that I'm going to show it as an example today is when I do this thing called passaging with my cells, we will sometimes take cells that are growing on a plate and we'll want to put them on a new plate, but we need to put them at a very specific density. So in order to figure out the density on the new plate, we use the C1 V1 equals C2 V2 formula.
So as an example from the lab, if we want to put our cells onto a 12-well plate, we will need a total of 12 milliliters, or 1 milliliter per well, and this will be our final volume, 12 milliliters, or V2. And the final concentration that we'd want to use in this example is 7.5 times 10 to the fifth cells per milliliter. That will be our C2. We can figure out our C1 by using an instrument in the lab to calculate the concentration of cells per milliliter, and then we basically solve for V1 to figure out what volume of cells we should use to end up with the final concentration and volume that we need. So for this example, you can see that we end up needing 6.0 milliliters of our original cell suspension, that's our V1, and we have to add an additional 6.0 milliliters of media to end up with 12 milliliters total, which is our V2. Then we just take that 12 milliliters of diluted cells and put it on the cell plate.